Now, it's my honor to invite our next speaker, Dr. M. Gouri Devi. Ma'am is director of Gouri Hospitals. She's director, Ridge IVF Group, vice president, fertility preservation. Ma'am doesn't need any introduction. We all are here because of teachings from her. She's a teacher of teachers. She's president IFS, vice president NACHI, member executive board at AOGD from 1994 to 2014 multiple times. She's State Quality Assurance Committee, Government of NCT of Delhi. She's a member at MTP Advisory Committee, has received many awards, and we all are looking forward to a oration from our great orator, Dr. M. Gouri Devi. Ma'am, please.
Thank you very much.
It's all yours.
Can I share my slides, please?
Yes, ma'am. Please. Welcome, ma'am.
Can you see that?
We can see,
Yes,
ma'am.
ma'am. We can.
All right. First of all, before I start my talk, I would like to congratulate Anuradha for an excellent lecture. It was very good, Anuradha. My topic today, do I have half an hour or should I cut short?
Ma'am, you have half an hour.
Okay. So, my talk today will be genetics for the clinicians. Those clinicians who are around, we have heard so much about genes. I wonder whether we will get any normal patient at all. So many genes Anuradha was quoting. Anyway, human genetics in the last century has advanced so much. And most of the diseases, all these monogene disorders, SNPs, etc. Besides that, even your normal diseases like obesity, diabetes, hypertension, cerebrovascular, Alzheimer's, etc., even they have got a sort of a genetic component. And the markers which we are doing have increased to millions. Now, whole genome sequencing can help pharmacogenomics. Maybe a time will come when the pharmacogenomics, every patient is given a chip and the whole genome is sequenced, and you know what drugs will help this particular individual in a disease, what time that some disease will prop up, what drugs are more effective, what drugs she will be or he will be reactive to. That is not a far off this thing. Already things are moving. Now, to date, it is possible to easily scan association between genetic markers by way of genome-wide association studies. I won't talk much about it because from morning till now, we have been listening to great scientists. The four main genetic disorders: single gene inheritance, multifactorial genetic inheritance, chromosome abnormalities, and mitochondrial genetic inheritance disorders. We heard about DNA, how the double strand is there, how the proteins are there, how the chromosome. I will not go into the details, but it was, the DNA was first recognized by Misher in 1869, as far back as that. And it is after 1 century that Watson and Crick described the double helix structure. Now, the DNA in human, it is about 2.2 meters, which is coil. That is what is DNA packaging. The clinician should understand. It's a DNA packaging means about 2.2 meters of the DNA. It is packed into an extremely compressed and compact structure, which is called chromatin, and that is pushed into the nucleus. Now, it could be monogene or Mendelian mitochondrial disorders, something related to protein where there is mutants. It usually happens with us, when it could be oxidative stress, it could be chemotherapy or radiotherapy, so on and so forth. And cytogenetic disorders where number, structure, deletion, duplication of the chromosomes, etc. Now, for us to know, what are all the platforms we have for genetic analysis? People have been talking from morning. I'll just put it to you for an easy thing. Conventional karyotyping, FISH, microarray, that is the CGHS, array, comparative genomic hybridization, SNP array, and quantitative PCR, and then NGS that has come under that picture. Now, conventional karyotyping is for dividing cells only. I, as a clinician, will not go into the details of these, what we do is a peripheral smear. From that, you arrest the cells in mitosis. From there, we extract the chromosome, and the report that comes will be like this. And a genetic person can put it in the proper perspective as haploid and tell us that yes, there is a trisomy at 21, as you can see here. That is how the karyotyping is done. Now, here, what are the advantage? You get a global snapshot of the entire genome. It detects both numerical and structural aberrations, but and mapping of the gene location. Only problem is it is limited to dividing cells and it is cumbersome, and there is an issue with the detection of mosaicism. Now, fish or the fluorescent in situ hybridization. Morning, Dr. Das was talking. You are labeling it with fluorescent dye, denature and hybridize, and they use locus-specific probes. And what you get is you can find trisomy 21, trisomy 18, translocations, and very rare syndromes like Engelmann syndrome, Di-George syndrome, etc. What are the advantages of fish? Specific detection of chromosome, and main thing is very fast. 2-3 days, your report is ready. Ease of detecting mosaics, and there is no need for dividing cells. But the disadvantage is we should have a prior knowledge of the chromosome involved so that we can use the proper probe like Down syndrome or a Philadelphia chromosome syndrome, as in AML. It is not useful when abnormalities are unknown or poorly defined, as in the case of solid tumors, spontaneous abortions, etc. And it cannot be applied for whole genome explorations. Array CGHs, you isolate the DNA, patient and control, you have fluorescent labeling in different colors. Then they go into hybridization, array, fluorescent signal, microarray scanner, and software interpretation. That is how that comes in. Now, coming to the PCR, already Dr. Das has said, you have to denature it, later annealing it, then extension with the addition of nucleotides, one at a time to the growing end of the DNA strand using the parent strand as a template. Now, the quantitative real-time PCR determines the actual amount of PCR products at a given cycle. Amplification is monitored in real time and quantified through intensity of fluorescence. Then high resolution is there. Now, sequencing, that is the NGS or next gen sequencing, high yielding DNA sequencing technique. Here, what happens? It is a massive parallel sequencing. It allows to sequence and annotate genomes at a very fast rate, and thousands of sequencing is done. An enormous amount of data is accumulated with the NGS sequence. Now, generally, there are three steps. As clinicians, we wouldn't know the details of it, but what they do is to extract the DNA, do a library prep, then amplification with the using PCR, then sequencing is done by several approaches. One of my previous speakers spoke about it. You can do either a single gene PCR, you can do a targeted panel sequencing, or you can do a whole genome sequencing or whole exome sequencing. That is WGS and WES. Now, what are the applications of this NGS? Targeted panels can identify known sets of gene mutations like thalassemia. Clinical exomes can discover new mutation in known genes associated with disease. Whole exome and whole genome, they can discover new mutations and genes only coding region associated with diseases. Now, choice of the technique, you can, if it is I think we have gone through it right from the morning, you can just read through it. And then we come to the important things of pre-implantation genetic testing. As clinicians, we use it. It could be PGT-A, SR, M, or PGT-P, which is for polygene disorders. In male, there are various, my previous speakers have told, cystic fibrosis, chromosomal anomalies such as Klinefelters, Y chromosome microdeletions, and so on and so forth. 2 to 8% of male infertility causes are due to genetic problem. In Klein-5 test, you have an XXY. What is the importance? The importance is if you can early detect it, some of these mosaics, we are able to get semen with spermatozoa, which can be cryopreserved and can be used for the person's biological child. And some of them, if you do a micro-TC, you might be able to find sperm, real good sperms, which we can use, and PGT of the embryos is recommended. Then in Y chromosome microdeletions, they have in detail talked CFTR mutation, where there is a bilateral absence of vastif, both the ways are there. In female, it could be a Turner syndrome. Again, what is important is if it is a mosaic Turner, early if we detect, we might be able to, at that time, sometimes when we catch them early, they might be still ovulating. And if we can preserve their fertility, we can do ovarian stimulation and take out the oocytes, we can do it. But most of the time, by the time they come us, there is no, and in a real exo, we won't be able to do it. Fragile X is associated with POI, and from Anuradha, we know that there are so many other genes which can cause PII. We were all the time thinking of a Fragile X. Then congenital adrenal hyperphagia, which can affect both men and women. And it changes in the adrenal gland can cause hormone imbalances, which can lead to change in the menstrual cycle, which will affect the fertility. Then chromosomal rearrangements, like whatever, pericentric inversion, translocations, this can increase the risk of pregnancy loss or result in an offspring with birth defect or intellectual insufficiencies. Now, coming to PGD or pre-implantation test, you have M for monogene disorders, SR for structural disarrangement, aneuploidy, PGTA. I will not talk much about PGTA because you have got an oration coming up after this. So in the genetic diagnosis, it is for monogene disorders and structural rearrangement. So for PGTM, either parent is deceased or carrier, sufficient evidence from the family pedigree, already affected sibling is there, so you have to do it, or previous miscarriage or stillbirth with a pathogenic mutation. In detail, they have been talking autosomal recessive, autosomal dominant, X-linked recessive, and X-linked dominant conditions from morning. These are all the different common single limb disorder we very often come across with Becker muscular dystrophy or spinal muscular dystrophy, beta thalassemia, cystic fibrosis, Fragile X syndrome, hemophilia, so on and so forth. Now, here I would like to talk about HLA typing because it is often asked to clinicians. There is a thalassemia major baby with the couple. Now they want to know, can we produce another sibling who will be able to give bone marrow or cord stem cell transplantation? So they want a normal baby. There we have to, if we can do an IVF, make the embryos, and those embryos can be tested for HLA typing. Yes, this is possible, and we can give them hope. Now, structural abnormalities, you have reciprocal translocation, Robertsonian translocation, inversions, and deletion, duplication, etc. Just for my clinician friends, what is a reciprocal translocation? You can see here, there is the breakage. So when this gets attached here and this gets attached here, that is a reciprocal translocation. Whereas if you have a Robertsonian translocation where the break is there, instead of this getting attached there and that here, what happens is these two gets attached, and that is a Robertsonian translocation. Then you have inversion. You can see the DEF is inverted here and attached there. So these are all the structural things that can happen. Now to diagnose, you can use either a FISH, a HCGS, or NGS. PGTA is actually analysis of a chromosome copy number, and it ensures transfer of a euploid embryo. And why we do it? It is to increase the live birth rate, decrease early pregnancy loss, and also reduce the time. Usually the indication are advanced maternal age, elective single embryo transfer, history of recurrent miscarriages, RIF, or severe male factor. Now, what are the sources of biopsy? As clinician, we are wondering, what are the sources? Polar body, cleavage stage, trophectodome, blastocell fluid sampling, and spent culture medium sampling, which people are talking about. Polar body, the disadvantage is you are doing the biopsy of the polar body, which will only give you about the maternal error can be detected. The post-zygote errors we cannot. So that is only where we are doing. So mostly what we are doing are either a blastomer or a trophectoderm. Now, blastomer is on day 3 at the 6 to 10 cell. 1 or 2 cells are just taken from there, and you use a laser to make a hole and just pull out 1 cell, and that is done. Now here, advantage is it detects both maternal and paternal. Fresh embryo transfer is possible here, but it is invasive. And what we do, when I mean is fresh embryo, from day 3, you take it to day 5, and you can transfer in the same cycle. It is an invasive procedure, and 1 or 2, your mosaicism can lead to, sometimes even good embryos are destroyed thinking it is a mosaic. In a trophectoderm, you do day 5 embryonic, ideal situation, 5 to 10 cells are sampled. You use a few pulses of this thing, laser, take out from the outer layer. We are taking it out from the outer layer. Inner cell mass is not disturbed, and at least 5 to 10 cells are taken. So you have a good, larger number is available. Mosaicism might be detected. Disadvantage is chromosome makeup may not be representative of the inner cell mass. And usually these are not fit for a fresh transfer. We freeze them and transfer in the next cycle. Now, non-invasive PGTA, here from day 3 to day 5, when the embryo grows, that fluid is taken for the analysis. And some people, what they do is taking it out from the blastocell. That is not a much preferred thing. So the spent culture medium is used, but again, ESHRE says 2023, they have not completely permitted it. The first NIPGT baby was born on March 2016. This is what it is. For origin of false positive mosaicism, sensitivity is 82, specificity is 84%. Now, is cell-free, this was a systemic meta-analysis came in 2020, low amount of the DNA in SCM, contamination by exogenous or maternal DNA despite careful denudation. So weakens the diagnostic efficacy of SCM for DNA for PGT. So that's again, further studies are needed. Concerns with PGTA, mosaicism, false positive, and embryo damage. Now, what do we mean when I say mosaicism? Mosaicism, this is usually, it occurs due to segregation of chromosomes. It results in an embryo which has a mixture of diploid and aneuploid cells. And what can happen with a mosaic is that there is either no pregnancy or a biochemical, can have a miscarriage, birth of a mosaic cell with birth defects and mental retardation. Having said that, you can have a healthy child with a live birth of a chromosomally normal child. Why? Self-correction of mosaicism is reported, and also inaccuracy of the initial embryo biopsy. Embryo damage. According to current evidences, embryo biopsy unlikely causes enough damage to embryos to affect the implantation rate. It is generally accepted that trophectoderm biopsy has less impact on the embryo viability compared to the cleavage stage biopsy. So, to summarize, PGTM for monogenic disorders, qPCR, NGH or ACGH, post-SR structural arrangement, use FISH, ACGH or NGS, PGA aneuploidy, NGS, and these indications I have already mentioned. Now, source of biopsy also I have covered. Now, coming to 1 or 2 recent advances, what is a 3-parent baby? The technique is used as part of a donated egg to replace disease-causing faulty mitochondria. The child would inherit DNA from a third person, and that's a mitochondrial donor, whose genes could be inherited by a future generation. Then has come the very controversial gene editing, genome editing. Genome editing has been plant and animals, this was going on because there is no ethical or legal issues. The CRISPR-Cas technology is a versatile genomic engineering, and as soon as it came in, in there was a lot of hubbub and excitement in the international biomedical community. Then came, this was published in Nature 2019. Janke from China, he recruited couples, HIV positive couples, and he edited the CCR5 gene, and twin girls were born. And he went and published it. And not only published, he presented it in an open gene editing conference in Hong Kong. He was much criticized. He was arrested, put in the jail for about 3 years. And now he has come out again. He is planning to work on gene editing. And those two girls are well, and they are going to the nursery school. So to conclude, the field of genetic offers clinicians tools for diagnosing, treating, and preventing a wide range of genetic disorders. It's essential to do this with sensitivity and caution, considering the legal and social implications. As genetics advance rapidly, collaboration between clinicians, patients, and reproductive biologists is essential to translate the genetic discoveries for individuals and communities. Genomic medicine is a reality that is already changing the management of rare diseases, cancer, and pharmacogenetics, and has enormously developed to guide the development of new drugs, transform healthcare, and improve the population health. Thank you very much for a patient hearing.
Thank you, ma'am. That was really a complete talk, starting from the very basics to covering the even advanced aspects. I'm really, really thankful. And I am very sure that all the audiences learned a lot. We do have certain questions, but I am sorry, because of shortage of time, we'll be taking it at the end of this session.